Hi, everyone. Thanks so much for joining us today. My name is Bill Isaki. I am the GDC User Services Team Lead, and I'm here with my colleague Gajanan Ganji. He is the GDC Data Quality Engineer. And today,
Hello all, welcome to the webinar.
today we'd like to talk to you about the data quality that is implemented within the Genomic Data Commons. So just to go over a bit of an outline for the agenda for today's webinar, we're first going to have an overview of the GDC data lifecycle. This is the steps from which data comes into the GDC, and data is made available to end users of the GDC. Next, we're going to talk about, within that lifecycle, all of the data quality strategies that are used by the GDC. This will be a stepwise overview of all the checks that are used that happen, like I said before, from submission to release throughout the data lifecycle. And lastly, I'd like to go over some of the GDC data quality metrics that are used and the tools that are used internally and made available to end users. So, might as well jump into it. Like I said before, the first part of this webinar is the GDC data lifecycle, from submission to release. This is a simplified flowchart for how data enters the GDC. And the first step you can see on the top here is that the submitted raw data is uploaded to the GDC data portal and into the GDC database. These are, this includes biospecimen, clinical data, as well as molecular files, and metadata about those molecular files. Once data files are uploaded into and they are in the GDC database, they are harmonized using a series of bioinformatics pipelines. This is one of the main functions of the GDC, to process data depending on the data type through a series of standardized pipelines. And these are the bioinformatic pipelines that are referenced here. If you want more information on those pipelines, we have lots of documentation and a few other webinars that detail them in a lot of detail. Once these files are harmonized and we have processed data, that processed data is submitted back into the GDC database using some internal pipelines, and that data is then indexed so that it can be displayed in the GDC data portal, which is what you all see at portal.gdc.cancer.gov. Now we have checkpoints, and each of these arrows in this flowchart represent places where data is transformed or relocated. And these places in the flowchart are steps in which problems can be introduced to the data, whether it's some data being missing, data being transformed in some way, maybe incorrectly, or other problems like that. So that brings me to the next section of this webinar. This is a very quick introduction, and this is most of the meat of the webinar, is the GDC data quality strategy. This is what we do to ensure that high quality data is made available at the GDC. So each of these checkpoints provides an opportunity to verify that the data is in good shape. And we have the strategy broken out into 7 steps here. In future slides, I'm going to go over each of these in more detail, but just to give you an introduction, first is a data submission application. What it says on the box, it's an application for submitting data to the GDC. The GDC data dictionary that verifies that incoming data follows the GDC schema. The submission data validation, which verifies file size and checksum. Pre-harmonization QC, which is a verification that incoming data can be harmonized and there's no problem with it. The QC and filtering steps that are embedded in bioinformatics workflows, the pre-release data review that verifies that no obvious issue exists in the data, and release QC. This is the very detailed QC that verifies that the data we release is as expected, and of course the correct access level. So I'm going to keep kind of coming back to this very simplified flow chart here. But here are the steps that I just talked about before. And if it has a little person next to it, that means that this is a human or manually performed data quality step. And the little robot signifies that this is something that happens automatically. And as you can see, there's one of the steps that uses both of these methods. So getting onto it, we have the data submission application. This is what you will send to the GDC if you have data that you think fits in the GDC and you would like to submit. The application gives information about the study and the data that will be submitted to the GDC. This is basic information like what is the DV gap ID, what kind of cancer is coming in, what is the study about, who should we contact. Very basic things like that. The submission application is then assessed by a committee, and the committee will approve or reject the application. And this is what we use to determine whether or not a project is a good fit for the GDC. This committee, when they go through the assessment, this is based on three main criteria. And I believe we actually go through this in a previous webinar about the data dictionary. But basically, it's based on the subject of a study. So is it a cancer study? And does it have an accepted manuscript? How much data is there? Does the amount of effort used to make this data, or will the amount of effort used to make this data available to the wider research community be worth the value to the research community? And one way we assess that is how many patients the study covers and how many different experimental strategies are there. The third criterion here, the richness of data, is mostly based on how much clinical data is present. So a lot of molecular data for a lot of experimental strategies is, it's nice, but it's not that valuable without rich and a good amount of clinical data being associated with these molecular files. Moving on to the second step of the GDC data quality strategy is the GDC data dictionary, and this is a completely automated step. If you've ever used the GDC submission portal, you have used the GDC data dictionary. This is the schema that defines each entity being a case, sample, submitted align read, that can be submitted. So this just tells the user what the properties can be. Is it an enum, string, integer, number? And which one of these properties are required? One of the things that the GDC has been working on lately is migrating strings to enum values. And this is for better quality and security and to prevent PII from being submitted into a string. Because as you know, a string can be anything. So we've actually migrated a number of things, such as all of the different diagnosis codes, all of the different diagnosis names, and even all of the therapeutic agent or drug names. So if you check out the data dictionary, you will see a very long list there. If you have submitted to the GDC before, you will know that users cannot submit data that goes against the schema. So if you were to submit something that for an enumerated value that doesn't match against that enumerated value, it will throw an error and reject your submission and will only accept it once that error is corrected. Also, a part that actually isn't automated is the GDC data dictionary is updated once per quarter. We are in close contact with the groups that are submitting projects to the GDC, and we will make updates based on new valuable data that comes in that we think will be valuable for researchers. There's a lot more details about that in a previous webinar about the GDC data dictionary. At the bottom here, I just showed on the GDC documentation website where you can view the data dictionary, and I'm going to go over just a very quick view of that. So this is what the viewer looks like. This one in particular is for the demographic entity. As it sounds, this provides the demographic information about a case or patient that has been submitted to the GDC. And here are three of the many properties that exist within the diagnosis. So, sorry, not the diagnosis, the demographic. And as you can see, the property is named in the first column, then described with just a text description. And then these three happen to be enumerations. So they give you the values that can exist for this property. Some of these could also just say string or integer. Next is a column letting you know whether or not this is a required field. If it's required, it means that we actually need information about it, or we need the submitter to verify that they have no information about it using an unknown or a not reported value. Lastly, we have a link to an external database, which allows users to compare what the property means and what their enumeration means in other databases that aren't the GDC and how they're defined there. So the next step is submitted file validation. And this is a completely automated step. And another step where I will say if you have used the GDC submission portal and you have submitted files, you have been through this method. So before a user can upload a file to the GDC, the submitters are required to provide the file size in bytes to the file and the MD5 checksum metadata. So once the file is then uploaded, a script happening behind the scenes in the GDC will verify that the file size and the MD5 sum of the file actually match the uploaded file size and MD5 sum. This is to verify the integrity of the file. So, this will stop a submitter who perhaps uploaded the wrong file, or will also prevent things like corrupted files happening. So if you have a BAM file, you know the MD5 sum, it goes through the upload process, and it ends up different on the GDC's end. It might be a corrupted file, and will be likely unable to be harmonized. So this is a step that prevents too much time from being wasted, and lets submitters know right away that the file is incorrect. And this is reported under the file state field in the submission portal. So the next step is pre-harmonization QC. This one is also integrated into the submission portal. And this performs logic checks that go beyond the dictionary schema. So the dictionary schema will validate things on a per entity basis. So as long as the demographic is okay and the case is okay, both of those will be accepted. So an example here is if you're submitting a FASTQ and you upload a read group number that's not R1 and R2, and you have more than one FASTQ in the read group, it will throw an error and say, we're pretty sure this is paired end data. You need to verify that these files are R1 and R2. Another example is that for BAM submission, we'll verify that the read group information that the submitter provides in the read group metadata in the read group node matches the read group information in the BAM header. And this is to verify that the read group that the read groups that are being separated and aligned during harmonization are more or less as expected. Another very quick example is making sure that the experimental strategy is the same between a read group and submitted aligned or unaligned reads. So if you report RNA-seq in one place and whole genome sequencing in another place, the GDC will not know how to process that. So the pre-harmonization QC reports these on two levels. Critical errors, so the aforementioned RNA-seq and whole genome sequencing in the same sample, that would be a critical error. Harmonization absolutely cannot happen until this error is resolved. This is completely automated, and harmonization cannot be requested until these errors are resolved. We also have another series of another level of error, and that's the warning. If you have warnings in your project, harmonization can still occur, but the error should be verified to let us know that this is actually how you want your data. For example, if there are two samples within one aliquot, this looks kind of weird to us, so we'll throw a warning. But some people do pool together two different samples into one aliquot. We have observed this, so we won't stop harmonization because this is, it's okay. But it will throw a warning so that people can verify that everything is good. Within the data portal is the pre-harmonization QC tab. This shows the type of nodes or entities in which the pre-harmonization QC errors exist. So as you can see here, we have 9 in the cases, 6 in the aliquots, and 11 on the read groups. And then it also has a breakdown of whether the errors are on the aligned or unaligned reads and which experimental strategy they show up on. Then at the bottom is just a basic count of how many critical errors and how many warning errors exist. And like I said before, all critical errors must be resolved by the submitter before harmonization can be requested. So now I'd like to get into the bioinformatics QC. This is mostly automated steps that are implemented by our bioinformatics team. So one of the things that the GDC does is collect data-specific metrics in the harmonization workflow. So based on the data type, certain metrics are collected. One of the metrics here are bad quality indicators, and if these are bad enough, the data is completely blocked from release. So an example is cross-patient contamination. The GDC removes data if it has 4% cross-patient contamination, and this is to prevent any germline leakage from happening because the somatic calling pipeline is designed for there not to be cross-patient contamination. One of the things the GDC doesn't do is remove data due to some other error types that aren't as dangerous. Different submitters, different users have their own criteria, and some rare cancer projects, we want to preserve as many samples as possible. Um, what we like to do is expose these metrics, and users can decide what to use instead of having the GDC removing data that isn't perfect. Another thing the GDC does is fix data quality issues when they're found within a workflow. So, for example, um, variants are filtered by the software DetoxOG if the OXO-Q oxidation metric is at a certain level. Also, some TCGA whole genome amplification data were processed differently because there were some batch effects that were being noticed there. So another thing that the GDC does, and this is mostly with alignments, is that QC metrics are collected and exposed to users. So this includes things like total read counts, insert size, read length, etc. And I'm going to talk about this a bit more later down the line in the third section. There are also QC metrics that are collected for internal use. These are workflow-specific metrics, and these are collected so that the GDC can improve bioinformatics pipelines on an ongoing basis. So something I'd like to bring up is that quality metrics are also reported internally on some bioinformatics files to inform users, but not remove any files unnecessarily. These are automatically implemented within the software, and some examples of this are flagged in VCF files to classify variants, and those flags will depend heavily on which workflow is being used. Mutex, Muse, Semantic Cipher, they all have different flags. So those are present. Structural variation files have scores in them, which tells the user how many reads overlapped this particular structural variant. Within math files, we have a few columns within the controlled maths that report the number of reads that support each variant. So, of course, you have a mixture of tumor and some normal, and you might want to see how many reads support each variant, and maybe establish a cutoff value for yourself. And then open access math files go through a very strict data quality pipeline. And if you want more information about that, please watch the previous DNA-seq webinar that will be made available soon on the GDC website. It goes into lots of detail for how the open access maps are generated. Okay, so now I'm going to hand things over to Gajanan, and yeah, he'll talk to you about the next two steps between now and release.
Thank you, Bill. So once the data passes bioinformatics QC checks, the data is ready for release. The next step in the data quality strategy is pre-release data review. Pre-release data review is done by submitters to ensure data content in terms of biological meaning, and submitter approval is sought for the release planning. The GDC team also performs spot checks, especially when data is generated from new workflows or there are major workflow updates. Data summary reports such as internal MAV report at the project level is generated to facilitate such reviews. Next slide, please. Once the pre-release review is completed, next step is release QC. The release QC is a standard data release QC process. It consists of 3 sub-steps. The first sub-step is a feature test. Feature tests help to ensure only expected data is released to users and files have correct access level. Feature tests also include detailed checks to ensure each node in GDC database have correct values populated for each property and have correct version and release number associated with each node. When I refer node here, it could be a file node or it could be any other clinical node such as cases, diagnosis, or demographic node. Further, feature tests also help to verify data migration due to dictionary updates, such as changing value of a enum or moving a property from one node type to other. For example, on read group node, we have a property called platform, and at the moment, let's say it accepts lowercase Illumina value. However, the new dictionary update makes it a title case Illumina. In such cases, all the read group nodes in GDC needs to be updated to have title case Illumina on the property. So feature test also helps to ensure such data is migrated. Next slide, please. The next sub-step is a regression test. The GDC data regression test is a suite of automated tests that are run every release once the new data indices are created. The regression tests help to ensure no release files are removed unless they are versioned. Also, these tests identify data updates to the released node properties, which further helps to validate those updates are done with approvals, or else we have to go back and validate why the data is updated and reach out to the submitters and make sure that data is valid. The last step is to validate all the downstream files generated by GDC which are distributed to GDC data consumers. For example, release manifest, which is available on GDC release notes, has only expected release files in it. Any deviation from release expectations are investigated and resolved if necessary. Next slide, please. Let's move to the next section of the webinar, which is GDC data quality metrics and tools. At the end of release QC testing, a release QC report is generated. Release QC report summarizes the feature and regression testing performed for a given data release. The summary contains number of passed, failed, blocked, and total tests performed in both feature as well as regression test cycles, with a link of test cycles in the Jira tool. At GDC, Jira is used to manage the test cycles and defect tracking. Though GDC data releases only contain changes to the data or new data is being released, the regression test also includes other modules such as API portal, which are quite often affected by data changes. These tests ensure new data work with these modules and doesn't introduce any breaking changes. Next slide, please. The QC report also includes a summary count of files by experimental strategy and data format for each project under existing cases. It helps perform additional high-level validation to make sure only expected number of files are released. The initial count is a raw data import file count that is available for import. The blocked count column is a count of blocked files due to redactions or files being not yet ready for release. For example, those files are under pre-release data review. The next column, expected count, is initial minus blocked. This is the count of new files that are expected to be available in the new indices. The actual count is the actual count of new files found in the new indices, and the difference is calculated between actual and expected. Generally, the difference is expected to be 0. However, if it's a positive number, which suggests more than expected files are released and help to track down those unwanted additional files. On the other hand, if it's a negative number, which suggests less than expected files are available in the new indices, which again helps to track down which files missed from being released. Next slide, please. This table is similar to previous table, that is file counts under existing cases. However, this table represents summary count of files by experimental strategy and data format for each project under new cases. The distinction here is file count under new cases versus previous table is for existing cases. The reason for the distinction is when new cases are being released, these new cases are at different level of harmonization, which produces files with different experimental strategy and data format. Once these cases passes pre-release QC checks and submitters approve the cases to be released, files under those cases may have additional files with a different experimental strategy and a data format than that of already released cases. This segregation between existing and new cases helps us to track to release more smoothly and stop unwanted files being released by mistake. For the next set of slides, I'll let Bill take the control. Thank you, Bill.
Thanks, Jonathan, for giving us a look behind the scenes at the GDC. And now I'm going to go over some more user-facing things. So first, I'd like to talk to you about some of the aligned reads metrics that were previously mentioned in the bioinformatics section. So as I said before, a set of QC metrics are produced for alignments automatically, and these are directly imported into the GDC database and then eventually available through the GDC API. So as an example, on the right, I retrieved a set of BAM files and displayed their contamination level and total number of reads in a JSON format. So as you can see, we have the file name, the file ID, as well as the contamination, which all happen to be 0 in this case, which is good. That's a cross-patient contamination, by the way, and the total number of reads that are aligned in the alignment. This is also available using the API in like a TSV format. So I would encourage you to look at our API documentation and API webinar if you are interested in doing that. I also would like to let you know that these QC metrics, we started producing them a couple years ago, so they may not be available for some of the older files, and that includes TCGA. However, when reharmonization does happen sometime down the line, these values will end up being populated for some of the older alignments. Here are some examples of the aligned reads metrics. These are most of them, and I don't have time to go through every single one of these for you, but I will focus on the ones that I've highlighted in bold here. That's contamination, MSI score, MSI status, and total reads. Contamination and total reads I just went over in the last slide, and that is contamination is the percentage of reads that are thought to be from a different patient than expected, so cross-patient contamination. This is what we use to determine whether or not a file is blocked from release, since data release shouldn't happen if cross-patient contamination reaches a certain level due to the risk of germline leakage. Total reads, like I said, is the number of reads that are aligned in a specific alignment. And then the MSI score and the MSI status are both values that come from an external piece of software called MSI sensor. The MSI score is reported as a number, and the MSI status is reported as a category, MSI or MSS. As a step, as the GDC dictionary, that was one of the first steps in the GDC data quality steps. It can be kind of a complicated thing to use, especially when migrating data over from another likely very complex project. So the data dictionary search tool was developed to ease this process. This allows for a dynamic search of the GDC dictionary, and very usefully, it assists in mapping external properties and values to the GDC schema. So what I showed you before was the GDC dictionary viewer, which is helpful, but only gives you exactly what's in the GDC dictionary. And the search tool is a bit more dynamic, like I'm about to show you. So the way to get to this is to go to the GDC documentation website, go to the Data Dictionary tab, and click on the search link. So let's say I'm a user and I have some very rich data about tobacco smoking. So I want to know how the GDC can accept this data in its dictionary. So I would type smoking into the field, the main field for the data dictionary search tool. And once a search happens, you can see we have 3 tabs here: values, properties, and dictionary. Values shows the enum values that match to the word smoking. So as you can see, we have 2 here. They both come from the follow-up entity, and one is from the comorbidity property, and another, specifically tobacco smoking, is reported as a risk factor. Clicking on the next tab here is the properties tab. This brings up a few more values, and this is actually the number of properties that match your search term, in this case, smoking. So we have a few more. All of these happen to be on the exposure entity, and we have things like tobacco smoking status, which is a categorical classification of whether or not someone smokes, doesn't smoke, or used to smoke at different levels. We have smoking frequency, how often somebody smokes, and the onset and quitting year of somebody who did smoke tobacco. So all of these are available as properties and can be explored in detail by either visiting the data dictionary viewer or clicking on the see all values under the property, under the 4th column there. A bit less useful, but good if you would like to explore the data dictionary, is the dictionary tab. This allows you to see where these where fields are located in the context of the entire dictionary, and together where the properties and enum values are located. So now, going to another example. Let's say I have a cancer project. It's been approved for submission to the GDC, And I have some primary diagnosis values that I need to submit to the GDC. But I don't know if the words that I call the cancer are the same words that the GDC uses. And it's quite unlikely that everything's going to match, including, capitalization, the way that words are reported, underscores, those kind of things. So there's a function here. So I know which property I'm going to submit to. It's the primary diagnosis under the diagnosis node. And we have a function compare with user list. Me being the user, I would click on that. And then let's say hypothetically, and I'm sure usually people would have much more complicated values than this, but I just put my defined values as breast cancer, colon cancer, and lung cancer. At the top, I can select whether partial matches or synonyms are okay. And once I had all my values there, I would click the compare button on the bottom. And this would bring up a comparison between the user-defined values and the matched GDC values. And as you probably guessed, there's usually a one-to-many relationship that happens here because in this case, I gave a very general term, breast cancer. But this can actually, as I'm showing here, be exported as a CSV, which can be viewed in Microsoft Excel. So in the first column, we have the user-defined value. We have all the matched GDC values for that user-defined value. We have some external database codes that might be useful. So for example, the ICD-03 code and the NCIT codes. And then it gives some more synonyms to show how this value was actually matched to the user-defined value. Lastly, I'd like to show off a submission data validation tool that is found in the GDC community tool page, and this is the read group tool. We found that during data submission, submitting read groups was causing some issues for many submitters. So a strategy was developed to simplify this. And the purpose of this tool is that if you're a submitter who has a BAM file, this tool automatically extracts the read group metadata and provides you a TSV or JSON file that can be submitted to the GDC as a read group. This will also verify that some of the pre-harmonization QC checks are successful because the pre-harmonization QC steps check against the BAM from your read group data, and this read group data would come directly from your BAM file. So if we have any submitters here who are looking to start submitting read groups, this is an easy way to do it. So I would encourage you to check this out on the GDC Community Tools page. That's the end of this webinar. I would just like to thank everyone for joining, and I have some GDC references here that might be useful, including the GDC website, which is a high-level documentation site, the GDC documentation site, which is more of a user guide, the GDC dictionary search, which I described in detail a bit ago, the GDC community tools page where you can download the read group tools. And as always, if you have any questions that weren't answered in this webinar or you can't find on the documentation, feel free to contact the GDC help desk at support@nci-gdc.datacommons.io. I would also encourage anyone who's interested to join the GDC user listserv, which can be reached by clicking on the support tab at gdc.cancer.gov and clicking here, join the GDC user listserv. And this will let you know whenever there will be a new feature release, a new data release, or if the GDC might be down for something like a maintenance window. So thank you all so much for listening to this. I want to let you know that there will not be a GDC monthly webinar in December, and the GDC wishes you all a happy holiday. Thank you so much.
Hey, thanks, Bill and Gashnan. We do want to take some questions, so if you have any questions, please enter them now in the Q&A section of the WebEx. I see there is one question. Are the GDC bug-related warning error issues resolved?
I'm not sure what that's referring to.
Yeah, could additional details be provided in the question on that? I will say that we do, when a defect is detected and reported to the user services, we do try to address them in releases. And when we do a release, we do put the release notes on the GDC documentation site. So we do have a list of known issues and issues that have been resolved in releases as well on our doc site in the release notes. And that kind of item related to warning issues would be in the GDC data submission portal release notes. But can the submitter of the question provide some additional details on that? Okay. While we're waiting for some additional questions, I did also want to point out that if you go to our GDC website, which is gdc.cancer.gov, and if you select publications, and if you do a search for GDC publications, there's a really good publication. It's called Before and After, a Comparison of Legacy and Harmonized TCGA Genomic Data Commons. And what it did is basically there was a comparison done between the TCGA data sets that were harmonized based on HD19 and our GDC we harmonized based on HD38 reference genome. And it's a really good article that goes through the different data types and discusses how they've done the analysis, and they did indicate that there is a high concordance between the HD19 version and the HD38 version. So there's a detailed publication in Cell Systems that you may want to take a look at if you're interested in additional some quality control checks that were done by the GDANs, the analysis working groups. Okay, let's see if there's any additional questions. And this video, it was recorded and it will be on our GDC website under support GDC webinars. It does take us time to process the videos, but they will be on there, in the near future. Okay. I don't see any additional questions. That being said, thank you all so much, and you all have a happy and safe holiday. Thanks for your time.
Thank you, everyone.